Okay, so welcome back to this next video in which we are discussing embryonic and induced pluripotent stem cells. Okay, right. So we're currently in the process of discussing transcriptional control of gene expression, and the reason we're doing this is because we want to understand what is it that confers specialization on a cell. Okay, what is it that keeps a neuron and keeps a hepatocyte a hepatocyte? Okay, we've discussed that the reason that they have such different phenotypes is because they have different transcriptomes and different proteomes. They are expressing different genes. But why is the question? What controls the gene expression in those two specialized types of cells and keeps the gene expression appropriate for that specific type of cell, i.e. to create a cell of that phenotype? Okay, that's what we want to know. Okay, now to answer that question, we are looking at epigenetics, okay, and the most important part of epigenetics is the transcriptional control. Okay, so we've seen that there are these things called transcription factors, which are proteins which can bind to specific sequences on the DNA called cis-regulatory sequences that are usually between 4 and 10 base pairs long. Now there are two types of transcription factors: general transcription factors and specific transcription factors. General transcription factors, you have very few of those. Okay, they're the ones that are required to bind to cis-regulatory sequences in the promoter regions of all genes, and they form the foundations for the assembly of the RNA polymerase II multi-protein complex on that promoter region, and then the RNA polymerase II can work its way along the coding strand and synthesize a piece of mRNA. Okay, specific transcription factors, which are the ones we're more interested in this discussion, are also called transcription regulators, and those bind to cis-regulatory sequences in the gene control region upstream of the promoter region, so the portion of the gene control region that's upstream of the promoter region. Okay, and we've discussed that they form these complexes usually, and this complex can either activate the formation of the RNA polymerase II on the promoter region or repress it. Okay, so this is how they can influence gene expression in a huge number of different genes, basically. So these transcription regulators, it's worth saying that they won't just bind to the gene control region of one gene. You know, this cis-regulatory sequence, you'll find it in the gene control region of a huge number of different genes, and therefore they can influence the expression of many, many genes. Okay, right. So we're currently in the process of discussing how do these complexes of transcription regulators actually affect the probability of the RNA polymerase II assembling on the promoter region. Okay, and we've discussed this quite direct way that you can do it. Okay, either the complex of transcription regulators can actually bind to the RNA polymerase II, or indeed the general transcription factors themselves, and influence the probability that the RNA polymerase II is going to assemble there, either towards activating the assembly or repressing the assembly. Or slightly more indirectly, they can bind to the mediator complex, and then the mediator complex can bind to general transcription factors or the RNA polymerase II and either activate or repress the assembly of the RNA polymerase II there. Okay, right. However, another really important way that transcription regulator complexes influence the expression of the downstream gene is by altering chromatin structure. Okay, so let me explain something to you here. So basically, we've discussed that DNA is not just free in the nucleus. Instead, it is wrapped around these histone core complexes like so. And in fact, most of the DNA is wrapped around histone core complexes. A relatively small fraction is in the linkers in between. Okay? Now, if the DNA is wrapped too tightly around these histone core complexes, then basically that any promoter regions in that sequence that's very, very tightly round the histone core complex is not going to be available for the binding of the general transcription factors and then the binding of the RNA polymerase II. Okay, So, one of the key ways that you can activate the assembly of an RNA polymerase II complex on a promoter region is by loosening the DNA around histone core complexes, and you can do this through histone modification. So this is another way that transcription regulators can affect the probability that RNA polymerase II is going to assemble on the promoter region. Okay, so let me just show this. So I'll firstly start by redrawing out that picture. So let's have our DNA here. Okay, let's have our gene again in blue. Okay, like so. Then we've got our promoter region here. Okay, and we'd quite like to assemble the RNA polymerase II on our promoter region here. Okay, but at the moment, let's say that this promoter region is in a portion of the DNA which is wrapped very tightly around a histone core complex, and therefore isn't available for the binding of the general transcription factors and the RNA polymerase II multiprotein complex. Okay, so the way that this portion of the gene control region upstream of the promoter region can promote the assembly of RNA polymerase II then is by loosening how tightly the DNA is wound around that histone core complex. So what can happen then is that you can get the assembly of some of these transcription regulators here. Okay, so here are these transcription regulators which will all have bound to their own little cis-regulatory sequences in this region of the DNA here. Okay, and now what these can do is they can bind to enzymes which are going to modify histones. Okay, so depending on whether this complex of transcription regulators wants to increase the expression or decrease the expression of this gene, it's going to bind to different enzymes. So let's say it wants to increase the expression of the downstream gene, then it might bind to an enzyme called a histone acetyltransferase. So I'll draw this here. Okay, right, so this enzyme at the moment is going to represent what's known as a histone acetyltransferase, or a HAT for short. Okay, so this stands for histone, that's the H, and then the A is for acetyl, and then the T is for transferase. Histone acetyltransferase. Okay, right, so what these enzymes do is they put acetyl groups onto components of the histone core complex, and what this leads to is the DNA ending up winding round the histone core complex less tightly. So basically the DNA loosens up around that histone core complex, and this can make available this promoter region here for the binding of the general transcription factors and then the RNA polymerase II. Okay, so histone acetyltransferases put acetyl groups onto the components of the histone core complex, and this opens the DNA up. Okay. On the other hand, if this transcription regulator complex wanted to repress the expression of this downstream gene to decrease the probability that the RNA polymerase II is going to assemble on this promoter region, then it could bind an enzyme known as a histone methyltransferase, an HMT. Okay, so this time stands for histone, that's again the H, and then the M this time is for methyl, and then the T again is for transferase. And basically these enzymes transfer methyl groups onto the components of the histone core complex, and this causes the exact opposite to transferring acetyl groups on. This causes the DNA that is wrapped around that histone core complex to become tighter wrapped around that histone core complex, and therefore effectively closes the DNA and absolutely makes sure that the promoter region is not going to be available for the binding of the general transcription factors and then the RNA polymerase II. Okay, so those are two other important ways that transcription regulators can influence how likely the RNA polymerase II is to assemble on the promoter region of a gene, and therefore how likely the downstream gene is to be transcribed. Okay, so it's not just through these direct interactions that determine the probability that the RNA polymerase II is actually going to assemble there. It's also through this chromatin remodeling by modifications to the histones by which these transcription regulator complexes can work. Okay, right. So the key thing to take from this, as far as we're concerned, is that these transcription regulators, they can change gene expression. Okay, they control which genes are going to be expressed and which are not. And the other really important thing that I'm going to come back to is the fact that it was the combination that was important. Okay, so it was a combination. Combinatorial control was the way that it was called, but we'll just call it combination control. You were controlled by the combination of transcription factors that you have, okay, because remember, it's the complex that is formed that's important, not just the individual transcription regulators, okay. So when you mix transcription regulators together, that can create very different effects on gene expression than you would expect if you just summated the effects of all of them, basically. Okay, so it's more complicated than that. It's the combination that's important. Okay, right. So now let's talk about the big question that we've been building up to this entire time, which is what determines the specialization of a cell? What is it that controls which genes are being expressed in a specialized cell type and therefore controls the phenotype of that cell? Okay, so what's important now is this concept of master transcription regulators. Okay, so there are a huge number of transcription regulators in human cells. Okay, a subset of these regulators are what are known as master transcription regulators, and these master transcription regulators are the ones which determine cell specialization. Okay, and it's these master transcription regulators that we are going to turn our attention to now. Okay, so these determine what cell type you are going to be, basically. Each cell type, each specialized cell type, will have a set of master transcription regulators. This is the basic concept. Every single cell type, so be it a hepatocyte, a skeletal muscle cell, a neuron, a cell of the pancreas, every single specialized cell type will have a set of master transcription regulators which it has expressed. Okay, and these will control everything, basically. They will determine which genes are expressed and which are not. Now you might ask, well, do they literally then bind to the gene control regions of every single gene in the genome? Well, no is the answer. Okay, what they do is they work through downstream transcription regulators. Okay, so let me explain this. So you have this combination of master transcription regulators expressed in your cell, and this determines what cell type it is. Now, how does it determine this? Well, basically, the idea is that these master transcription regulators will then alter the transcription of a bunch of genes, and then the bunch of genes that they do alter the expression of will then, many of them, will also be transcription regulators. Okay, so the idea is that these are going to create downstream regulators. They're going to control loads and loads of downstream regulators, and then the downstream regulators are then going to control actual protein expression. So they're going to control more widely gene expression. Okay, so the big problem here was, how can just a small subset of transcription regulators control everything? Well, the way that they do it is they work by controlling the expression of a load of downstream transcription regulators, and then those downstream transcription regulators, their jurisdiction spreads more widely, basically. And this is how these master transcription regulators can control the gene expression of pretty much every gene in the cell. Okay, right. And influence then the phenotype of the cell. So this is going to cause the phenotype of the cell. Okay, so to go over this again, every single specialized cell type will have a set of these master transcription regulators which are expressed. So maybe I'll put these here. So for this cell here, it's got a set of master transcription regulators. Okay, one here is in red, one here is in blue, one here is in purple. This is the set, and usually you'd have a few more than this, although we'll see that the master transcription regulators for a pluripotent cell, such as an embryonic stem cell or an induced pluripotent stem cell, there are just 3 of those. Okay, and we'll come to those in a moment. Okay, right, so every cell has this set of master transcription regulators, and what do these do then? These go in and influence gene expression. Now, they don't influence the gene expression of every single gene in the genome themselves, okay, but they influence the gene expression of many genes, okay, so of a subset of the genes, and of course they work as a combination, so the combination is important, combinatorial control here, okay. And then overall what that now does is it alters the gene expression of firstly some proteins directly, but it also alters the gene expression for many proteins that are actually transcription regulators themselves, and we'll call these ones downstream transcription regulators because their expression is going to be controlled by this set of master transcription regulators. Okay, and now the change in expression of all of these downstream regulators, they are now going to control loads of different genes basically. So this is how you can amplify the effect from just these 3 master transcription regulators. These can change the expression and determine the expression of a huge number of downstream transcription regulators, and then those downstream transcription regulators, they can then influence the expression of the entire genome, and that's how you control the entire genome's expression level, the epigenetics of the entire genome, just by this small set of transcription regulators, which are the master transcription regulators. So this is how these control the entire phenotype of the cell. Okay, so basically, this is why all the different cell types are different. All of them have a different combination of master transcription factors or master transcription regulators expressed, basically. Okay, and you might think, well, isn't this laughably simple? Do you really think that's the way that specialization of cells is completely controlled just by a set of a few proteins? Okay, but when you think about it, it makes sense. Why wouldn't development use such a simple system? Okay, it's got the huge challenge of orchestrating the cells in the right place to become the right type of specialized cell. So wouldn't it be nice for it to have a very simple way of determining what a cell actually becomes? So this may seem like a laughably simple system, but maybe this is actually how it works, that nature has this incredibly simple way of determining what cells actually become. Okay, and then the ridiculously complicated thing is about making cells in the right place have the right combination of master transcription regulators so that they become the right thing. Okay, so although it may seem laughably simple, maybe this is the way that nature works, that it has this very simple scheme where to become a certain cell type, all you have to do is express the right combination of master transcription regulators, and they then influence through this amplification effect of the downstream transcription regulators the expression of all genes in the cell, basically. Okay, and therefore determine the cell's phenotype. So that's the central concept of this all, that you start with these master transcription regulators, they amplify through downstream transcription regulators to spread their control over absolutely all genes in the genome. Okay, right. So let me now say a correlate of this, okay, or a corollary of this, rather. Okay, so a corollary of this is that we can transdifferentiate cells. Okay, we can take a cell of one type and turn it into another type by changing the combination of master transcription regulators that it expresses. Okay, and this process of turning one type of specialized cell into another type of specialized cell is a process that we would have once thought was impossible. Okay, 100 years ago this was impossible, but we can now do it, and it's called transdifferentiation. And all you have to do is change the expression of these master transcription regulators. Okay, so let me give you a nice simple example of transdifferentiation, although it's worth noting that we can do this now for many different conversions. Okay, so a nice simple example is you can convert fibroblasts from the skin. Okay, now fibroblast is a cell that's involved in creating connective tissue. Okay, so it secretes things like collagen. Okay, so you can take a fibroblast from the skin and you can turn it into a skeletal muscle cell. Okay, now this is actually a very simple conversion to do, and the reason is that fibroblasts are actually very closely related to skeletal muscle cells in development. Okay, so in fact it turns out that in the fibroblast the combination of transcription regulators, master transcription regulators that you have, is the same as the combination that you have in skeletal muscle cells minus 1. Okay, so if I draw a very simple picture of this, and I shouldn't draw a fibroblast like that, I should actually draw a fibroblast in the shape that they are. Okay, so fibroblasts are in this sort of spindle-like shape here. Okay, here's its nucleus, and I'll draw its master transcription regulators, like so. So again, we'll just draw 3. I don't know how many you actually have for a fibroblast. Okay, but here's one in turquoise, here's another in blue, here's another in red. And basically, that combination of master transcription regulators in the fibroblast is the same as what you have in skeletal muscle cells minus 1. So you're only missing 1 master transcription regulator. Okay, so if we draw this in the skeletal muscle cell, okay, we can have 4 master transcription regulators. 3 of them are exactly the same. Okay, so here's the turquoise one, here's the blue one, and here's the red one. Okay, but there is one additional master transcription regulator that is in skeletal muscle cells but is not in fibroblasts. Okay, so all you have to do is put in this final master transcription regulator that's missing from the fibroblasts, and in fact then the fibroblasts end up converting into skeletal muscle cells. Okay, their gene expression changes so much that their phenotype ends up changing into a skeletal muscle cell. Okay, so what is the name of this missing transcription regulator here, this master transcription regulator that we need to put into the fibroblasts in order to convert them into skeletal muscle cells? Well, it's called MyoD. Okay, so if you put that one missing master transcription regulator into a fibroblast from the skin, you can force it to convert into a skeletal muscle cell. Okay, so that's a rather incredible corollary of this. Okay, right. The next thing I want to just talk about is why then, when specialized cells divide, do both of the offspring cells, the 2 daughter cells, remain the same specialized cell type? Okay, well this should hopefully be extremely obvious, but let me just spell it out. Okay, so the first thing to say is that certain fully differentiated cells cannot actually divide. Okay, so in particular, muscle cells, myocytes, both in the heart and in the skeletal muscle cells. Okay, so I should put probably striated myocytes. So skeletal muscle cells and cardiac muscle cells and neurons. These are examples of fully differentiated cells, specialized cell types, and they can't divide. Okay, and because they can't divide, they are what are known as terminally differentiated cells. So if you ever hear someone talking about terminally differentiated cells, all it means is a differentiated cell that can no longer actually divide. Okay, so these are terminally differentiated cells. So cell division isn't a problem that we need to even consider in these types of cells. Okay, and this is actually why cancers of muscle cells either in the heart or in the skeletal muscle, and also tumours of actual neurons are extremely rare. Okay, usually when people talk about brain cancer, it's not the neurons themselves that have actually formed the cancer, it's usually the glial cells. Okay, because they have this huge lock on cell division, it's very unlikely that a somatic cell will get enough mutations that allows it to then break all of the restraints that are placed on it to actually develop into cancer, okay, in a normal human lifetime. Potentially, as our life expectancy goes up and up and up, we may start seeing more of these cancers develop, okay, when you're living for longer. Okay, right. So those are terminally differentiated cells. They can't divide, basically. However, other specialized types of cells are capable of dividing. So let me give you some examples. So fibroblasts, these connective tissue producing cells, these are capable of dividing. Okay, hepatocytes within the liver, these are also capable of dividing. Okay, smooth muscle cells, which I'll just abbreviate down to SMCs, these are all capable of dividing, okay, in a normal human adult body. And when they divide, the two cells that they produce are the same specialized cell type. So let me show a hepatocyte here. Okay, so when it divides, it produces two more hepatocytes. It doesn't produce a hepatocyte and then suddenly a smooth muscle cell. Okay, it produces two hepatocytes. Now, why does the specialization get passed to both cells? Well, we can answer that easily now with our master transcription regulator theory. Okay, our master transcription regulator theory would say that this hepatocyte has a certain combination of master transcription regulators, which I'll just colour in as coloured dots here. So let's say it has the orange one, the purple one, the red one, and the blue one. Okay, and it has loads of these expressed. Okay, and then when it divides into two, then both of the daughter cells will end up with some of these in their cytoplasm, and therefore they will still have that expression, basically. Okay, they'll still be the specialized cell type. Okay, so here's one of them, and then the other will have the same combination of master transcription regulators. And I've actually missed out an incredibly important point about these master transcription regulators that I'd just like to go back to now, okay, which is the self-perpetuation. Okay, this is really important. I've just realized that I've forgotten it there. Okay, right. So, basically, I've said that every single specialized cell type has this combination of master transcription regulators which determines its entire gene expression via the influence on the downstream regulators. Okay, now what I haven't said is how does the cell maintain this combination of master transcription regulators? Because, you know, these are proteins. They will eventually get degraded as part of the normal proteostasis mechanisms. Okay, so you need to continually be producing more. Okay, and the way that this occurs is because these transcription regulators here, the master transcription regulators, they're capable of inducing their own expression, basically. Okay, so each one of them will activate its own genes expression, basically. So this red one here will be activating its own genes expression, the blue one will be activating its own genes expression, and the purple ones will be activating its own gene expression. And the combination is actually also capable of activating each other's gene expression. So for instance, the blue and the purple one are also capable of activating the red one's gene expression. Okay, so basically the idea here is that this combination of master transcription regulators, not only does it influence the expression of every other gene in the genome, okay, but also it influences the expression of its own of the genes of its free members, basically, and it maintains their expression at a high level. Okay, so that's the concept here of self-perpetuation, that it maintains itself. Okay, and that's why you don't suddenly get a specialized cell type changing to a different type of specialized cell, and also why this can work Okay, because you can see that both of these cells will now have lower concentrations of the master transcription regulators, but now they can just repopulate themselves basically in these two daughter cells because they can activate their own expression basically. Okay, and that's how you can transmit the specialization down to your daughter cells. Okay, if you're a specialized cell that is capable of dividing, i.e. you're not a terminally differentiated cell. Okay, right. So now let's turn our attention back to developments then. Okay, let's go back to differentiation of the embryonic stem cells or the inner cell mass cells into specialized cells. So remember, these inner cell mass cells here, they were capable of producing any cell of the adult body. Okay, so what is that process? What is specialization? What is differentiation? What has to happen to turn an inner cell mass cell into a neuron? Well, all it needs to do is acquire this appropriate combination of transcription regulators, of master transcription regulators, and then they will take over the gene expression of the cell and turn it into a neuron basically. Okay, so that raises the question, is it the case that these inner cell mass cells, these pluripotent cells then, is it the case that they have absolutely no master transcription regulators expressed? Are they a clean slate basically, that all you now need to do is introduce master transcription regulators into there and then they'll gradually evolve, or I shouldn't say evolve, change into a more differentiated cell until you finally add a large enough combination that it becomes one of the specialized cell types? Okay, well the answer is no. They do have master transcription regulators of their own that maintain them as these inner cell mass cells, these pluripotent cells, or if we're talking about the ones in culture, as embryonic stem cells. Okay, and basically this combination of master transcription regulators that is responsible for the pluripotent state, what it does, what it ensures, is that all the other master transcription regulators that are involved in the differentiated states and involved in conferring a differentiated state, it ensures that all of those are suppressed, that you don't express any of the master transcription regulators which are involved in turning you into a specific type of cell. Okay? So that's the role of these master transcription regulators in these embryonic stem cells or inner cell mass cells. Okay, so what I now want to talk about then is what are these master transcription regulators that are present in these pluripotent embryonic stem cells, and then we can turn our attention onto what induced pluripotent stem cells are. So I think I'll go over the page for this because I don't think I have room to put it there. Okay, so the four master transcription regulators of embryonic stem cells or of inner cell mass cells that confer this pluripotency are, and these are very famous transcription factors, OCT4, SOX2, and KLF4. Okay, so these are the master transcription regulators of embryonic stem cells or of inner cell mass cells, and they confer pluripotency. They keep the cells in this pluripotent state where they have not gone into any of the differentiated states yet. And like other master transcription regulators, they are capable of self-perpetuation. Okay, so what I firstly want to look at is the self-perpetuation network. Okay, so how they activate each other. Okay, and then what I want to talk about is some of the downstream transcription regulators that they activate the expression of, and how those are going to confer some of the characteristics of embryonic stem cells and of inner cell mass cells. Okay, right, so let me first talk about the self-perpetuation network. So I'm going to draw the four in a triangle. So here's KLF4 at the top here. Okay, because that regulates the expression of absolutely every single one of them. Here is OCT4 and here is SOX2. Right, so every single one of these, KLF4, OCT4 and SOX2, all of them bind to the gene control region upstream of the promoter region of their own genes, okay, and therefore influence their own expression, and generally their pro-activation, basically. Okay, so they all bind to the gene control regions upstream of the promoter region of their own genes, basically. So let me colour this in. So we'll have KLF4's binding in orange. We'll have OCT4's binding in turquoise here. And we'll have SOX2 here in blue. Okay, right. KLF4 also binds to the gene control region upstream of the promoter region in both the OCT4 gene and the SOX2 gene, okay, and therefore influences gene expression for both of those two, okay, like so. Okay, OCT4 and SOX2 don't have any influence on KLF4, okay, they don't bind to the gene control region upstream of the gene for KLF4, but they do bind to each other's gene control region upstream of the promoter region, okay. So OCT4 binds to the gene control region for SOX2, and SOX2 binds to the gene control region for OCT4. Okay, and this combination of these three master transcription regulators promotes the expression of all three of these genes. So the KLF4 on the gene control region for KLF4 activates the expression of KLF4, okay, to keep KLF4 high. The SOX2, OCT4 and KLF4 that are all on the gene control region for OCT4 are going to overall, as a combination, upregulate OCT4, okay, and the same for SOX2, basically. Okay, so this is how you get that self-perpetuation. This combination of these three master transcription regulators maintains their own expression, basically. Okay, now what I want to turn my attention to is looking at some of the downstream regulators that give these embryonic stem cells that we have in culture some of their properties. So what were the key properties that we saw? The pluripotency and the fact that they can proliferate indefinitely. So I'm firstly going to start off with the proliferation. So KLF4 is capable of binding to the gene control region and increasing the expression of another transcription regulator called c-MYC, which is a majorly important transcription regulator if you're studying cancer. Okay, it basically binds to the gene control regions of a huge number of genes and makes the appropriate changes in gene expression that allows a cell to proliferate, basically. So it's a very, very powerful pro-proliferative transcription regulator. Okay, so c-MYC is then going to cause the proliferation. So this is why embryonic stem cells or inner cell mass cells proliferate a lot. Okay, so that's very helpful. And then let's look at another key downstream regulator. Another key downstream regulator is a transcription regulator called NANOG. Okay, and in fact all three of these master transcription regulators, KLF4, OCT4 and SOX2, all of them bind to the gene control region for the gene for NANOG. Okay, so all of them binds there, and the combination of the three then elicits a positive increase in the expression of NANOG. Okay, so NANOG is going to go up because of these three binding to its gene control region. Okay, right. Now, what does NANOG do when you create more of it? Well, NANOG is responsible for firstly controlling the expression of many genes which then produce the phenotype of a of an embryonic stem cell or an inner cell mass cell. Okay, so usually they are quite round cells, things like that. Their phenotype, their actual physical characteristics. So NANOG is responsible for many of the phenotypic features of these cells. Okay, and also NANOG is majorly important in preventing the expression of master transcription regulators that confer differentiated states. Okay, so inhibiting other master transcription regulators which are important in differentiated states. Okay, so for instance, MyoD was one of those master transcription regulators which we saw was in skeletal muscle cells. Okay, that's one of the ones that was associated therefore with a differentiated state, and it will have its expression inhibited by NANOG. Okay, so inhibiting master transcription regulators for differentiation. So this is why these embryonic stem cells will remain in the pluripotent state, basically, because they're inhibiting the expression of the master transcription regulators for differentiated states. Okay, so if you now want to differentiate one of these embryonic stem cells or one of these inner cell mast cells into a more differentiated state, you're going to have to override this and force the expression of one of these, or a combination of these, master transcription regulators for a more differentiated state. There's then going to be able to turn these master transcription regulators, KLF4, OCT4, and SOX2, for the pluripotent state off, basically. Okay, right, so we've now seen what it is that controls the specialization of cells. We've seen that it is this combination of master transcription regulators. In addition, we have seen the specific combination of master transcription regulators that is needed to maintain a cell in this pluripotent state, like an embryonic stem cell. Okay, in the next video we'll discuss what induced pluripotent stem cells are, and then we'll discuss some applications of this sort of research.